Hello, my name is Chris Corzette, and I'm excited to share a project called BiomePx with you. I'm working with a fantastic team here at the Alfred Mann Institute for Biomedical Engineering at USC, and our goal is to help convert next-gen sequencing insights into clinical diagnostics by developing a qPCR-based platform for targeted microbiome profiling. We believe this qPCR-based platform has the potential to accelerate biomarker validation and help bring microbiome research into the clinical practice. But to understand why we're so excited about a qPCR-based platform, I think it's helpful to first take a step back and address one of the fundamental challenges we face when thinking about the future of microbiome-based diagnostics. And that's the fact that we need a better way to efficiently quantify key bacterial biomarkers within complex microbiome communities. Next-gen sequencing has been an excellent method for discovering the preliminary correlations we're all so excited about, but there are significant limitations when thinking about applying NGS for follow-up validation studies and eventual clinical implementation. Not only is it fairly expensive and relatively slow, but most studies still rely on 16S, which often lacks the meaningful species-level taxonomic resolution demanded of microbiome-based diagnostics. Furthermore, next-gen sequencing presents unique challenges for standardization and reproducibility. On the other hand, qPCR remains comparatively cheap, sensitive, and fast, but it can be extremely challenging to identify primers that are specific for target bacteria. This is especially true when considering we're often looking for just a few significant organisms within complex communities that may have hundreds to thousands of other organisms that must be accounted for. So with that said, why are we so excited about a qPCR-based platform? It's because we've developed a bioinformatics pipeline that's actually pretty good at identifying taxon-specific primers capable of quantifying these meaningful organisms. Here, on the left, I'm illustrating just one example pulled from the literature where the relative abundance of a handful of genera were associated with an important clinical outcome. After analyzing the relevant taxa, we plugged them into our bioinformatics pipeline to identify primers specifically targeting each genus. We then start the process of assessing each candidate primer pair to ensure specificity. In these gels, each lane reflects template genomic DNA from one representative within each target genus, and each gel reflects a distinct genus-specific primer pair. In each case, we're only detecting a product on the intended target genus, illustrating the absence of unintended cross-reactivity. Furthermore, although not shown here, we also find this specificity holds true even within complex communities from donor stool samples, indicating these primers are suitable for downstream qPCR applications. Accordingly, we're in the process of developing a variety of qPCR assays suitable for targeted microbiome profiling that offer a variety of advantages. First, our bioinformatics pipeline allows us to tailor our primers to the specific level of taxonomic resolution for each bacterial biomarker. It allows us to walk up and down the phylogenetic tree to obtain the desired level of taxonomic specificity. We're showing genus-specific products here, but we can get down to the species or strain level resolution as needed. Second, while we're currently working on a few specific assays, our pipeline can be applied to a wide variety of microbial targets as needed for different clinical insights. As more putative biomarkers are uncovered, we can quickly develop customized panels for each application. And that's why we're so excited about this qPCR-based platform. We think it can really accelerate biomarker validation and clinical implementation. That's because qPCR offers a faster, cheaper, scalable platform for targeted microbiome profiling that can make it easier to validate exciting preliminary findings. Furthermore, once validated, a qPCR-based diagnostic will be easier to implement within clinical labs that are already familiar with the qPCR-based assays. Ultimately, BiomePx offers rapid, targeted microbiome profiling that can be deployed for a variety of research, clinical, and industrial applications. We're always eager to discuss new opportunities, so please feel free to reach out to either myself or Sapna Jane, the BiomePx project manager. Thank you.